So thanks very much for the invitation. Sheila, very much happy to be here and look forward to walking you through some of the work that we've done. So I'm sure you're all familiar with our very iconic species of whales. I'll start out with the orca or the killer whale. We have a number of different ecotypes, and an ecotype is defined as by basically its feeding group and its culture. Vocalizations are different between the different ecotypes. And there are 3 on this coast. We have the offshore, which you probably haven't seen. They don't come in very often. Bigs or transient killer whale. Those are the marine mammal eaters, and the resident killer whales. And I tend to focus on the resident killer whales. Those are the fish eaters with a primary desire for Chinook salmon. So they're a species that is, or a population that is endangered. So that's where a lot of my research is focused. And we really want to look at those human-based impacts and figure out ways to abate those threats such that the population will hopefully increase. There's another population of residents which are up in Alaska, but we won't be talking really too much about them today. So the really interesting thing is the northern and southern residents, as you can see from the range, they have overlapping ranges. They both feed on Chinook, but they have very different population trajectories. So the northern residents and the southern residents were both harvested for the live aquaria industry in the 60s. So there were a large number of whales taken and a lot that died during those captures. And they estimate about 60 some odd southern resident killer whales were removed from the populations. And they were choosing, they didn't want the large males, they didn't want the large females. So they really took the millennials. They took these young whales that really were the next generation out of the population. And so what happened was when they started doing the census, they did that through photo ID, a technique that was developed by Dr. Michael Bigg, and were able to identify each and every individual within the population. By 1974, they were able to do a complete census on the southern resident killer whale population every year since 1974. The northern residents, they don't see everyone every year, but they're able to keep really quite good track of the numbers. So as you can see, the southern resident population is languishing. It really has not increased. It got up to about 98 and then declined concomitant with the Pacific coast-wide salmon decline. And so both the northern resident and southern resident populations went down, but the northern residents seemed to recover from that, and the southern residents never did. So another real challenge for the southern residents is they are in poor body condition compared to the northern residents. So when we look at a whale, it's really hard to tell how fat they are. But with the advent of drone imaging, it's been much easier, because what colleagues of mine started in helicopters and then moved to drones, and they would basically take morphometrics, the entire measurement of the whale, and figure out what the best option was for a quick and dirty kind of index. And that turned out to be the eye patch ratio, because they basically start to get a neck and they start to get a divot in behind the eyes when they get really skinny. They call it peanut head. And so the ratio of the distance between the top of the eye patch to the bottom of the eye patch, if it's really far apart, they get a large ratio, and that means they're quite fat. And as they get skinnier, that ratio starts to get lower and lower and lower. So we can use that eye patch ratio to monitor the condition of animals over time. So southern residents are not in great shape. So that's another reason why we're really focusing on it. And so that's part of my role is to put the best available science on the table so management decisions can be made for things like supporting the recovery of killer whales. And 3 primary threats for killer whales really are contaminants, because they have a large blubber layer. So well, hopefully they do, but they have a blubber layer where contaminants are stored, and they eat Chinook salmon, which are quite high in contaminants. So they end up getting and maintaining quite a high load of contaminants through what's called bioaccumulation. And vessel traffic and disturbance. The southern resident killer whales spend probably 4, 5 to 6 months of the year in the Salish Sea. So they range from all the way up to north of Haida Gwaii and all the way down to Southern California, but they spend a disproportionate amount of time in these waters, in the waters right off the coast of Vancouver Island. And the reason for that is because their primary food source is Chinook salmon. And of course, the Chinook salmon are migrating during the spawning season back up to the rivers like the Fraser and into Puget Sound. So the killer whales basically set up camp for 3, 4, 5 months of the year. Unfortunately, that's right in the shipping lanes. So you can see in this figure, you can see the gray lines are the shipping lanes that service both the Seattle, Tacoma, and the Vancouver port. There's a lot of vessels moving through there. And of course, any urbanized area like Vancouver or Seattle has a lot of recreational vessel traffic as well. Whale watchers, ferries, there's a lot of noise in the ocean. And the problem with noise for killer whales is they echolocate. So they send out a signal and they need to listen for the return in order to see how far away that fish is. So when they start diving down to pursue a fish in deeper water, there's not a lot of light down there. They don't have very large eyes. They're not really a visual predator. They're using those echolocation pings to recognize and follow the fish. Now you bring in all this noise and they can't hear the return. So in fact, there might be a lot of fish in the sea and there might be enough food available for them, but they can't actually see it in those noisy sound clouds. So there's a lot of negative aspects to being a southern resident killer whale in this environment. So what I like to do is so much of this research through Dr. Michael Bigg and Dr. John Ford is ecological and really looking at whale behavior, identifying all the populations. And so now we're moving forward. We've got a really good handle on that. And I'm really interested in the biology and the physiology of these whales. And I'm sure you can imagine that's a very difficult thing to do. We go in, we get a blood sample. They could tell you everything that's going on. You go for a scan. We can't do any of that with the whale in the wild. So we need to get really creative and think about our different types of techniques. And we look at different matrices. And so a matrix is, your blood is a matrix, feces is a matrix, respiratory vapor is a matrix. We look at those different matrices because each of them tells us something different. So blood tells you immediately what's going on in the body. Blow or respiratory vapor, it gives you the metabolic picture of what's going on right now. When you look at fecal, it's about 24 to 48 hours. It tells you what happened in the last couple of days. And then biopsies, that can be weeks, months, years, depending on the storage and mobilization of fat. So I like to look and, if I can, get what we call the trifecta and get fecal, blow, and biopsy from the same individual because it allows me to have a really good picture of the metabolic history of the whale. So we collect a lot of poop. We float around behind whales. It's a lovely job, and just wait for them to defecate. And this is where master students come in really handy. This was a lot of work was done by my master student, Kate Yaley. She was in the field for 4 years. It was a very long master's, collecting fecal samples. And she did some of our seminal work that was really interesting on hormones. So what she did was she looked at something called thyroid hormone, which I'm sure most of you are familiar with. When you're healthy and you have a lot of energy, your thyroid level is quite normal. If you are in a situation where, like if you're a fasting animal or if you're a hibernating animal, you want to turn down the thermostat. You want to slow down your metabolism. So those types of animals will reduce their thyroid levels and become hypothyroid because it slows down their metabolic rate. So interestingly, if you look at the blue bar on the right-hand side, that's the southern resident killer whale fecal thyroid levels. They are much lower than the northern resident. And they're skinnier, they're not in as good health. And what's happening is they're either there's some reason, which is what we're trying to figure out, why they're thinner, why their body condition is poor, or they might have periods of food limitation. And when your food is limited, you slow your metabolism down. So it's like driving slower in your car to make your gas tank run a little bit longer. You want to sort of go at optimal. So they reduce their metabolic rate. And interestingly, the lowest of those individuals are the skinniest in the population. So these thin animals are saying, okay, I've tried everything I can. I've got to turn down the thermostat and slow down my metabolic rate so I can survive. So they're not doing that well. And so what we also do is we go in and we look at something called the metabolome. So in your feces is a history of your physiology that happened over the last 24 to 48 hours. And what we can do is we can look at each of these dots represents a metabolic byproduct. And what I do is I compare the northern residents to the southern residents. And what we'll see in here is anything in red is higher levels in the southern residents when compared to the northern residents. And you can see that even though they're the same species, they forage in some of the same areas, they feed on the same types of fish, they have significantly different metabolome, which tells me there's something physiologically different between these two populations. And so one of the interesting things that we looked at was something called succinic acid. It's a marker for inflammation. And in the southern resident population, it's significantly higher than the northern residents. They're also pooping out all their amino acids. You can see the 0 on the bottom of the graph. Let's see if I can point to it here, right here. Almost every amino acid is higher in southern resident fecal than in northern resident. And what's interesting about that is it's actually what we call a phenotype, or it's a similar kind of medical issue that you would see in someone who has Crohn's disease or inflammatory bowel disease. And the reason for that is because when there's inflammation in the gut, you start to get leaky channels between the cells and you start to lose things. They should be keeping their amino acids. That's part of the energy from the food. They should be retaining those and absorbing them, but instead they're pooping them out. So there's something going on in the southern residents that's not just about, oh, I don't have enough fish. It's my gut isn't healthy. And so one of the things you probably have heard about that's all in the news these days is your microbiome, which are all the creatures that live in your belly. And here's where we get into the mucky bits. If you have a fecal sample, the weight of it, 50% of the weight of it is bacteria, live and dead bacteria. That much is your microbiome. Like it's a really large amount. And what you really want to have is a really high diversity. You want to have a lot of different kinds. You don't want it dominated by a particular species. I'll show you on the far right in this image. You can see this is over time. I think the first sample was 2006, so day 0 of the study to day 5,000. You can see that the diversity in the southern resident gut is decreasing. In those fecal samples, it's decreasing because we have samples that from our colleagues down south that go back into the early 2000s. And so by assessing those, we can see that the gut is changing and not in a good way. And so this is something that's really been puzzling to us. So the fecal metabolome is kind of a map for me to be able to say, which one of these pathways would be affected by contaminants, which ones would be affected by food or prey availability, and which ones would be affected by stress or cortisol or disturbance. And what it does is it gives me a map to figure out how those threats are acting upon the physiology and making these whales less fit or making them less healthy. So this is my roadmap to science. And so this is what I use. So that's really talking about fecal. Now let's move on to blow. And one of the great things about blow is it happens all the time because they breathe. And so you can see there's just sample after sample that I can't get. And so this is where it's really, for me, I want to be able to crack the nut of getting blow samples because we call it blood of the wild whale because we can't get blood from the whale. And blood tells you what's going on in the body right now. But respiratory vapor tells you the same thing because it's what's going on immediately. So what we've been working towards, I just want to point out this fellow in the bottom here, he's constantly pooping. This one, you'll just see a steady stream of poop and they just tease us with samples all the time. So he's and then just because I love drone footage, you'll see there's a little one that hides underneath his mom. Let's see if I can just play that again. So this little guy there's a little tail you might be able to see just tucked underneath the mum. And then this is the one that's pooping all the time. There, see the little one pop out and come back in. So I think there's some really cool bioenergetics going there where the young ones get a little bit of slipstream, kind of like cycling, and you're in the draft of a larger whale. So it's kind of cool. So the problem with blow is that when you get a blood sample, you'll get things that, some hormone per mil of blood. But when a whale blows and we get that milieu of blow, the challenge is it's contaminated by seawater, and sometimes contaminated a lot, sometimes just by a bit. So what do we express the hormone level to? I can't say it's per mil of blow because half the blow might be seawater. And then, of course, we're flying or trying to get this blow, and some of it evaporates. So it becomes a real challenge. I was going to move forward to, so what I've developed is a technique that looks at a marker for seawater. And so I went and thought about all the components of seawater and said, well, what's unique? We looked at sulfate. So there's a sulfate component, which is the third. Our bodies have chloride, our bodies have sodium, but there should not be a biological signal of sulfate. So then what I can do is say, well, if there's sulfate there, I know that relates to the salt water. And it allows me to say, oh, how much is that sample contaminated? Is it 80% seawater or 20% seawater? So this is a technique that we're just starting out with. And it's been really, our initial bench studies have been really good because everybody collects blow and they run their assays and they say, oh, look, I can show that there's hormones in there, but unless you can quantify it, it's really not very useful. So this has been a major step forward in understanding it. So I just want to talk a little bit about blow in general. So we tend to use these fellows. So that's what I call the white mouse of the whale world. That's a humpback whale. And so humpback whales are, as you know, the populations are going way up. They're much larger and they don't have a dorsal fin. And that becomes really important when you're trying to collect a blow sample because there's this dorsal fin right behind the blowhole and we don't want to hit that. So the humpbacks are the species that we work with as we develop and test our techniques. Now you get an idea of the challenges of this and how large this whale actually is, because we're going to come up and try and do a blow sample. So you'll see us coming up with, just on the side here, with basically a popsicle stick. And we're trying to wave it through the blow as the whale exhales. So you can see how challenging this is. So there's like 4 Petri dishes on there and you're trying to get some blow on them. So this isn't really the best technique. So this we started to think about different ways in which we could collect blow. And we moved forward to, I just want to turn the sound off for a second, move forward to this. And this is our drone, and we've got this really cool little remote that allows us to open and close the Petri dish as it flies through the blow. And that really helps to, when you're flying around, the little droplets will fly out. So if we close the lid, we can now address the evaporation and we can address sample loss. So we fly this thing through and we close the lid and away we go. So this is just a video. So this is the drone, the video from the drone, and you see it's coming through and it just goes through, and then you'll see it'll say auxiliary light, and that means close the Petri dish. So this has been really helpful in improving our yield from the blow sample. So we're coming at this from a lot of different directions. And in November, this is 5 years in the making, we brought this phenomenal team of scientists together. We had the head of the Marine Mammal Commission in the US. I had 7 vets, cetacean vets from aquaria all over, and we all came together, and we're all working on blow in different species and all dealing with the same challenges. And we all came together for 3 days, and it was a phenomenal meeting. And from that meeting, we now have a collaboration where we're working with the whales in SeaWorld to validate the technique. So rather than using those precious samples from the field, we're going to be able to validate the technique in those individuals in the aquarium and then be able to take it to the field. And so we're doing this next month. And then this summer's field collection will actually yield valuable and quantifiable data. So it's a huge step forward. So you put a lot of minds in one room with the drone, it's a lot of fun. Okay, and then so the third matrix that we're talking about is our biopsies. So we use, we go through significant discussion on this, ethics approvals. We do not biopsy the southern resident killer whales. We have all the researchers have an agreed moratorium on that. And what we do is it's basically the size of a pencil eraser. So we send out what's called a bolt, which is basically an arrow, and then it has a float on it, and we pick it up at the end. And so you can see on the right-hand side, this is a biopsy. The black part is skin, and then the white part is blubber. And so we can get a lot of things from one of these. So we can do what's called whole genome sequencing. That's 2.88 billion base pairs. There's a lot of, A's and C's. And so it allows us to map out the entire genome of an individual. So when we biopsy an individual, we can then get the code and shorten that code to 9 base pairs. And we can identify each of we biopsied 217 animals. With just 9 base pairs, we can tell you which one of those animals it is. So it's been really helpful because then if I get a fecal sample and I don't know who pooped, I can actually tell you who it was because I'm able to go back and identify them. So this is really important, whole genome sequencing, something called methylation, and this is epigenetics. This is how the environment affects your genetics. And this is a really interesting field of study where we can start to look at how, for example, in the humpback whales, we have 2 breeding populations that come to our waters to forage. We have one from Mexico and one from Hawaii. And they come here and forage and they go back. Each of those breeding populations are subjected to very different threats along the way because they're very different waters. There's different shipping. One is along a coast, the other is out in the middle of the Pacific. And so by understanding what those animals are exposed to along the way and how that alters the genome, we're able to have a better understanding of the threats. We also look at things called stable isotopes to measure diet. And something really interesting we did is because we don't really like biopsying the whales, you don't want to do that unless you absolutely have to. We developed a live cell culture. And this is really important because it allows us to test things in the lab to look how contaminants affect them without actually having to work with the animals. So it's been a lot of trying to take the lab to the field, but then take bits of the animal back to the lab and see what we can figure out from there. So DNA, this is not your grandma's DNA. I used to do PCR and when I first started, and it was like a whole, it was a whole bench top. Now this fits in the palm of my hand. And this little tiny chip is, I don't know, you could just fit it in, easily. It's not much bigger than a toonie. And it's basically like a laboratory in itself. It's got a whole, it's microchip on the bottom, computer board on the bottom, and then you just inject the sample in and it basically runs what my entire bench top would run. So this is phenomenal. And what we get from this is we can obviously genetically see differences, for example, between the populations of killer whales. So if you see here, you've got transients, offshores, Alaskan residents, southern residents, and northern residents. So if you match up the colors over here, so if we see the pink is our offshores, southern residents and northern residents. And then if you go across, you can see that they're significantly different. So that it's a different color than what you would see from the northern residents. So you can see the differences between the populations. But what's interesting is when we run the whole genome for the humpbacks who come from Hawaii and come from Mexico, we don't see that. Even though we know they're breeding in those different areas, genetically, they're kind of mixed, which is interesting because they apparently breed and have their young on the breeding grounds and just come to our grounds to feed. So we thought, okay, somebody's jumping the fence here because there's definitely some exchange going on. So if we look at the female humpback whales, it's a bit of a continuum. So all these blue ones are from individuals that have been identified as a Mexican whale. And then if we look at the Hawaiian whales, you can see all the green ones on the bottom with some unknowns. And then all that in the middle is kind of a bit of a transition between the genetics of those two different breeding populations. And so I thought, how's that happening? So interestingly, there was a paper in 2022 that showed there were a couple of males who, during the breeding season, went from Hawaii to Mexico. And then another one who went from Mexico to Hawaii. So there was definitely some gene exchange going on there with the males. There were a few males moving back and forth. So I went to something called Happy Whale. And we upload all of our sightings and photos into Happy Whale, as do many, many researchers from around the world. And it's this phenomenal database that allows us to basically take our photos and of a very unique fin, and then there will be an AI matching that comes in and says, oh yeah, that's Puffer. Yep, he was last seen here. And so it maps out all the sightings. And so from our foraging area here, a lot of our whales are seen in both Mexico and Hawaii, but it's interesting when we look and see, there's about 2% of the males that go to both. So we know the males are moving back and forth. So this is Puffer. He's gone back and forth a few times, went to visit Mexico 1 year just for some picks. And this whale's been back and forth, seen 28 sightings on the foraging grounds, been back and forth. Wilbur just goes all over the place. He's a cosmopolitan fellow, bit of a Casanova. And so, but what's interesting is in 21,000 sightings, we only have 6 sightings of females that have been seen on both sites. So they have very strong site fidelity. This is their breeding ground. This is where they had their calf. This is where they go back every year. So I'm really interested in these ones that are seen in both locations. And so there's only, from these 6 whales, 5 of them have not been seen on the foraging grounds. They've only been sighted in either Mexico or Hawaii. But this one, Garuda, is really interesting. So her first sighting was in 1990 in Mexico. I think she was already an adult, judging by the photo. Like, it looks like she'd been around the block a bit. Her fin's a bit beat up. If you have a look, I'll show you the photo in a sec. And then she was seen 30 years later in Hawaii. And then her next sighting was Russia with a calf. So either she's a Mexican whale who suddenly decided to go to Hawaii, have a calf, and then go to Russia with it, or she was a Hawaiian whale who happened to be seen in Mexico in 1990. What's interesting with the humpbacks is, with many whale species, because you can kind of get away, sexual selection is female, right? Because you can choose who you're going to let mate with you, or you could just swim away. It's a very equitable world in the reproductive world of whales. So one of the thoughts from humpbacks is that, as with many populations, you're attracted to some aspect of fitness in the male, and it's often thought that it's the song. So I kind of wonder if she's a Mexican whale. I don't know if she speaks Hawaiian, but somebody liked her, because when she got there, she ended up getting pregnant and having a calf. So it was really interesting to think about how these females are selecting their mates, and how these males are being selected for by a female if they're not singing the song of the female's location. So I find this really intriguing. And just in case you were thinking that this was a misidentification, this is her from 1990, and this is her from 2002. There's no mistaking that fin. She's really quite beat up. Okay, let's move on to something called kinematics. I'm a metabolic physiologist, so I really like looking at energy flux in the whales. And because they spend 95% of their life underwater, I want to know what's going on there. So we use something called a D tag, or a suction cut tag. And these D tags are, just about this big. And what they do is they have a kinematic sensor in them that tells you the pitch, roll, and yaw. So the movement of that tag, and we can take those data and recreate the movements of the whale. In addition to those kinematic sensors, these 2 little things on the bottom here are hydrophones. So once we bring those data together, it's like riding on the back of the whale. We get the movements and we get the sounds. And so I'll just show you what that looks like. So when we tag them, we try and put it on the dorsal fin because we want to hear when they surface. So you can hear it, you can basically hear the go as it breaks the surface and as it goes down. And that really helps us to understand the surfacings, but it also there's an antenna on there, and we can find the tag with a Yagi in order to ensure that it's still on the whale. So here's a little bit of a video that will show you sound on these, Yeah, so these are, so it's pretty easy. It's not easy at all. Well, not easy at all. So suction cup, pull, boom, on the whale. Most of the time it goes well. Sometimes the whales do a little bit of shudder, and this is our biggest problem. They know exactly how long the pull is, and they'll come up just at the you're practically falling off the boat trying to tag them, and sometimes they'll be in a row, and you want the female, and she'll come up on the outside, and then you're ready. You're all ready to tag, and they come up, and then it's just someone else.
And what holds it to the fin?
Just suction cups. So it's just got these 4 suction cups on them. We can't see on this side, but see, there's just 4. Oh, and this is one of the most elegant bits of engineering. Do you see these little tiny white tubes here? There's a little metal loop on them, and there's a computer board in here, and we can program this tag to say send a current through that loop after 12 hours, and the loop will burn, and those little tubes will unfold, and they'll let air to the suction cup. And what happens when you put air under a suction cup? It releases. So it's such a beautiful, elegant bit of engineering, so simple yet so elegant. And so we can program the tag to fall off at a certain time, which is really cool. Okay, all right, so this is what all this technology, and I cannot change this picture to be a killer whale. It's a humpback, but this is a tag on a killer whale. And so what you're seeing there is the red pokes up is the upward beat of the fluke. The blue is downward. And what you see here in the green is the strength of that fluke beat. And so what you're seeing now is the whale's actually searching. I don't know why we don't have any sound though. Are you hearing clicking? Okay, they're not hearing it here though. Okay. Anyway, there's echolocation clicks they're going as they're searching, and then as they go down and chase and pursue the salmon, what you'll hear is crunching. There, I think a little bit there. Listen. Are you getting it there? There. Did you hear the crunch? So that's how we tell, okay, they actually got the salmon. Those are called prey handling sounds. And you'll hear a whole lot more. So he actually made contact and grabbed the salmon. And now as he comes up, look at how big these fluke beats are. He's getting to the surface because he's out of breath. I sped this up. This is up. Now listen here. Either ripping, crunching. So they're tearing apart the salmon because they prey share. So they will take part of it and they'll give part of it to another whale. So it's amazing. These D tags are incredible. So you can look at them and see the movement and understand the energy and calculate the energy required in order to forage. So this is really something I'm very interested in, is what is the cost of foraging? How do we figure out how much energy a whale puts out in a day or consumes in a day? And what's really important about that is, as I mentioned, we have skinny whales. So is it that they're not getting enough food? Is it they're having to forage more because of the noise and they're expending more energy in order to get the amount of the caloric intake that they need? So we wanted to come up with a few different ways to assess that metabolic rate in the field. So this is the great thing about science. We get to do MacGyver things and try and figure out different ways of taking the lab to the field. This is using an infrared camera, and I was trying to calculate the blow. Because when we talk about metabolism, it's actually measuring oxygen consumption. So we can count the number of breaths a whale takes, but you know, sometimes your breaths are shallow when you're relaxed, and sometimes they're very deep, so you're going for a run. So we need to know tidal volume. And so I thought, well, this would be great. If we can actually see the blow, we can measure the volume. So we built this this artificial whale. You can see him off on the side here. He's got water bottle lungs, and it was a lot of fun. So, and that's his blow coming out. It would be equivalent to about a 4-year-old killer whale in size. And so we validated this in the lab, but the problem is that when you go to the field, this is our blow from the lab, but this is the blow from the field. And the way an infrared camera works is it looks at differentials between temperature. And because that blow dissipates, the temperature is changing so rapidly in those molecules and in that water vapor. And then, of course, you can see the background makes a big difference too. So if something is on water versus the land, it kind of makes the edges fuzzy. So it was a good try, but it didn't really get us to where we wanted to be. So another method that we wanted to use was another way you can measure flow, like a garden hose, is if you know the diameter of the hose and how long the water is flowing, you'll actually be able to calculate how much water moved through it. And so we can do that. Here's our garden hose. We're looking at the diameter of a nostril. We can say, okay, this is how big the nostril is. And we say, how long was it breathing through that nostril? We can actually back calculate, we hope, to look at the volume. So this was an interesting study that was done that showed that that they changed the size of their nostril depending on whether they're breathing heavy or not. And so then what we did is we went through and as I just explained, you have a pipe, you know the diameter of the pipe, and you look at how long something is moving, the time it takes, the milliseconds of air moving through that pipe. You can calculate the volume. And so if you look here, you'll see here's the whale. We can do morphometrics to know how big it is. We can get an idea of what its lung size is. And then over on this side is the infrared picture. And you can actually, this is quite high up, but we can get quite a bit closer. And you can see the diameter of the nares or blowhole. And so we can actually measure that pretty easily with the drone. So that's a really promising method as well to try and get a handle on metabolism of the whales. All right, so that's energy out, but what about what comes in? And so that's really one of the big things about the killer whales having this very strong attraction to Chinook salmon is that many of the stocks of Chinook salmon are declining. And that's a bit of a challenge because obviously when you have a species at risk eating another species at risk, it becomes a bit of a moral game, I suppose. So energy the way energy is measured in a scientific form is we'll take, let's say, a fish and you chop it up and you put it in what's called a lyophilizer, freeze dryer. And then you take that freeze dried fish and you put it in a blender. And then you take a small sample of that and you put it in what's called a bomb calorimeter and you burn it. And when it burns, you measure the change in temperature and it tells you how much energy there was in every single bond of that fish. And so when you do that with a Chinook salmon, you get about 16,000 kilocalories, which means they would need about 17, 18 big salmon a day. Because killer whales don't just eat any salmon, they like the 4 to 6 year old, as the fishers do. So they like the big ones. The problem is that a killer whale is not a bomb calorimeter. They're picky. They don't eat the whole thing. They don't eat the bones. They don't eat the head. And so what we need to do is take that information and put it into the context of a whale in the field. And so you can see here, this is actually from a sablefish. They just sort of and just take the good stuff off and then leave the rest of it to float around. And so we have to try and understand that selective consumption. What parts are they eating? So if we say they need 17 whole fish, well, they might need 30 fish in the way they actually blends them out and partition them. So we want to get a better handle on how many fish they actually need to eat. What's really interesting is we're getting, from my colleagues, we had a killer whale, World Killer Whale Symposium last year, and it was fascinating, all the different ecotypes from all around the world. They have this selection. And so this is a researcher, Bella Reeves, and she's been bringing this all together to look at the different morsels that a killer whale will select from different species. So they'll just take the liver, or they'll just take the, the intestines. And what's interesting is, I thought this was a really interesting one. This actually was, the prey was still alive, and they went in and took the tongue. And so you wouldn't think that a tongue would be that large or nutritious of a meal. I'm just going to show you a photo that might be a bit offensive, but this was a humpback whale that died last October. One of the ones that was struck by a vessel just off in Howe Sound. This is its tongue, distended and coming out of its mouth. So this gives you an idea of how large a whale tongue is, a baleen whale tongue, because they push their tongue up to squeeze out all the water and keep in all the krill, right? So this is a really large organ that the whales are targeting. Okay, so yeah, we wanted to say, well, that's all different species. What about killer whales in BC? Do they have a particular type of salmon that they like? We know they like Chinook, but there's a lot of different stocks out there. And it turns out they are very picky. And this is a study I called What's on the Table. And so we looked at, from the rec fishing data, we said, well, what stocks are being removed from these areas? Basically, rec fishing is like a sampling. And so we can figure out, are you catching these Fraser River fish? Are you catching Vancouver Island fish? And then what we did is we collect prey samples. It's like crumbs off the floor after a killer whale's had a meal. We get our nets and we get the scales and bits of flesh. And we're able to do genetics on those and find out which stocks of Chinook they're eating. And it turns out they are very picky. And in fact, based on the availability, based on what's on the table, they tended to prefer the Fraser summer 4 sub 1s and the summer 5.2s. And what's interesting about those fish, I'll show you in just a minute. And then they didn't like broccoli, so they didn't eat their West Coast Vancouver Island and Columbia and some of the Puget Sound fish. So even though there's a lot of them, and interestingly, those are some of the hatchery fish, which, they would say, hey, we increase that to help with the killer whales. But the killer whales don't like those fish. And what's interesting as to which ones they like are the ones that are highest fat. No surprise there. And what's interesting in that is the highest fat fish are the ones who have to travel the furthest. So the Chinook that are fattiest are the ones who go all the way up the Fraser River. They need to keep those reserves because they're swimming up waterfalls and moving all the way up to, so you can see the spring 5.2 and summer 5.2 have the highest lipid content. They're the ones who have to go the furthest. And then the summer 4.1s. And if you think about Vancouver Island, so just here, if you see this little notch here, so Port Renfrew would be here. This is Bamfield. So the nit-nut fishery is here. They're basically a walk in the park. So those fish don't have very far to go at all, and they're not very fat.
What are the numbers for
That's a great question. It's salmon speak. It's so when they talk about a 5.2, that's a salmon that returns usually around 5 years of age, but spends 2 years in the river. So a 4.1 would be at sea for 4 years and 1 year in the river. So those fish that are what they call spring type, or stream type, that spend 2 years in those freshwater environments, they'll have a 2. And so the ones that are going to sea after 1 year, so the 4.1s, those are they only spend 1 year in. So in general, those spring type, those stream type salmon that have a long way to go are the ones with the highest fat. Those are the ones that the killer whales like. Okay, and in closing, so one of the things that we wanted to look at was we now have a good idea that they're eating the 4 to 6-year-olds. They like the fattiest fish. They like, these fish from these stocks. So we're able to map those out. So what we did, we wanted to look at the impact of recreational fishing because they've been closing some areas because of, potential impact to killer whales. We want to have a really good handle on what those impacts are. So I did a study called the Box of Whales. And this is using something called an agent-based model. What we did was we took a 25, 5 by 5 kilometer square box. And so basically it's like you're putting an aquarium in the water. And then we populated it with the number or the type of salmon in the area and the abundance that our colleagues in salmon had told us would be in this area. So we said, okay, we'll put fish in the box. We bring in a group of whales. So we have a general idea of the foraging groups. They range from 3 to up to 17 individuals in a group. And so we bring those individuals in. And so they're echolocating, they're finding salmon, and then we bring in the fishing vessels. So these vessels come in and they're producing noise at about 20 knots when they come in, and then they're fishing at about 2 knots, which also adds in sound to the environment. And it blocks those echolocation signals. So it's a really complex model that calculates the sound cloud, the distance to the salmon, the type of salmon, the desirability of the salmon, and it does it 480,000 different iterations as it goes through. But it's really helpful in having us understand 2 things. One is, what is the direct competition between a whale and having fishers remove the same fish that they're wanting? And the second is, what's the effect of noise of those vessels coming and going? So all of this leads us to coming up with a tool that allows us to help management understand those impacts and hopefully mitigate some of those impacts, but in a much more precise way. Because with species at risk, we tend to take a precautionary approach. So it'd be like, okay, let's close fisheries here. But now we're able to say, oh no, they only like these type of fish, and that's a August run. And in this area, we're okay, that area, you might want to do closure. So it gives us a much more fine scale tool to help advise management on how to mitigate these issues. All right, and in closing, obviously, I don't do this alone. I have a phenomenal team of researchers. This will be our 9th or 10th year going to the field, and couldn't do it without this amazing team, and of course, the whales. Thank you very much. If you have any questions, I'm happy to answer them. Oh, 8:30. Wow, I did it.
So, any questions from the floor here? Rush with this in your mind?
We're very specific about the Chinook salmon.
Mm-hmm.
How many Chinook salmon runs are there a year? Like, they must be eating something else in between.
Yeah, that's a really good question. So, as I mentioned, the way this research was initially conducted was by collecting debris, right? So in general, scales, like they glint, you can see them easily. And so what we realized is when we started doing genetics on the fecal and seeing the diversity of species, the technique might be a bit biased, of prey sampling might be a bit biased towards Chinook. Because, for example, sablefish or black cod, they have almost like a skin. So you don't get those pretty scales that are easy to see and collect. And also, it looks like they're hitting the juvenile sablefish, also a very high lipid fish. And they might just be taking them in one bite rather than sharing them. And they're, and when we see them foraging on sablefish, they sort of do this like almost like a pirouette. Whereas when they're going for a salmon, you see them looking and then down they go. And when they come up, you can smell it in the air, and they do this dorsal shudder and a victory lap. So there's behaviors associated with foraging on different types of like coho. They switch to coho and chum in the fall when the populations of Chinook finish their migratory runs. And then in the winter, their diet becomes much more diverse. And that's where I'm really keen on the energetics, because I think they have to really move around a lot, because they don't just get to sit there and their mouth open in the migratory path of the Chinook. They've got to move around a lot in order to meet their energetic needs. They come back in the spring skinny. So they are in the lowest body condition after that winter. So the winters are tough.
So they do learn to eat other salmon at least.
Oh, yeah. And I think that's, yeah, I think you eat what's most energetically dense and what's in the highest concentrations. So in this case, and also we talk about those ecotypes, you have these large top level predators and we've got 4 populations of them. But the way they can exist is I'll eat seals, I'll eat Chinook down south, you eat Chinook up north, and I'm going to stay offshore and eat sharks. And so you have that partitioning that leads to these ecotypes for survival reasons. So when we think about killer whales and think, why are you so stubborn? Just go eat something else, but part of that is deeply ingrained in them because it's what led them to survive by separating them out from other species, and the Chinook populations were plentiful. So will they start to maybe expand their summer diet? We'll see. And certainly we do see them, the fecal genetics is really helpful because it gives us a bit clearer picture of the ratios of different species in the diet, and allows us to understand a bit more than just the prey cycling.
So is it saying that it's an evolutionary adaptation, basically, of each of these whale types?
Yeah, I'd say a behavioral, yeah. So, I think that food is one of the food, shelter, and reproduction are the key drivers for any species. And you find your niche, you create your niche, and sometimes you get pushed out. And that's one of our concerns, is that the anthropogenic impacts are happening at such a rapid rate that the species, they live as long as we do, so to undertake these adaptations, it's really interesting. So some of the matriarchs, they lead the matriline to go to certain places, and they'll keep going down to Southern California where the runs have dried up, and they'll keep going into locations because that's where they went, and that's where those pathways and memories and behaviors were laid down at a time when food was plentiful in those areas. So we're seeing some interesting behavioral changes in the population with the loss of matriarchs. And so it's like, well, mom always came in here, and now we're seeing the southern resident killer whales come in far less frequently into the inner waters. So part of that is it looks like they're doing better in foraging in the outside of the Salish Sea. And there might be some aspects of events that's kind of really hard to tease out, but the loss of those matriarchs plays a role. And when you see the video footage, it's the matriarch who's leading. She takes you to where you go, and those are 60, 80, 100, maybe 100-year-old animals. And then so the environment, it's like your grandmother, the environment she grew up in is very different than what we grew up in. And technology is their anthropogenic changes are like everything we've seen in the last 20 years with AI or the internet or so it's rapid, much more rapid, I think, than many species have had to deal with in the past. So that's a challenge.
How much, you mentioned stablefish, how much stablefish is there like in our waters, and is the stablefish population stable? Is it
Yeah,
of course a popular food fish for people as
yeah,
well?
it's apparent. See, I actually don't eat fish.
More northern fish. I didn't even realize we had stablefish.
Yeah, they're so off of the if we go back to the La Perouse Bank, I'll just show you quickly a picture of talking about, or maybe not. No, we're stuck. Okay, so if you go out just of the mouth of Juan de Fuca Strait, so basically nothing between you and Japan, is the bank called La Perouse Bank, and that's where that's where the sable fishery is. When you look at Juan de Fuca Strait, where we talk about the whales migrating, or sorry, the whales following those migratory fish. Juan de Fuca Strait's really deep. It's about 300 meters deep. But then you come to something called Swiftsure Bank, and it's like every tidal cycle, this water comes and has to go up to about 80 meters. So you're taking all that biomass and all that energy and pushing it up onto this amazing, just phenomenal environment called Swiftsure Bank. It's like going down Juan de Fuca. It's like a bit of an aquatic desert, and then you get there and there's birds and there's humpbacks and there's killer whales. It's phenomenally dynamic because of that energy upflow or downflow. So we think that maybe they're eating the young sablefish that are coming up and being forced up the wall. Because sablefish are quite deep, and so there would be that energetic cost of going down to 300 meters to get a fish. So we're just trying to understand that. Where it's really challenging because how do we know? How do we, we could do co-occurrence. Here's the whales foraging, here's the sablefish, they're fat, they're full of lipids, we see it in the poop, so it's probably going on here. But it's really hard unless we do more tagging to get a better understanding.
So is it, sorry. Wanna, go ahead.
We have two
I'm
questions.
going to change the subject a bit, so this is a follow-up
Okay,
on this.
can we, before you
Yeah.
hold yours for a second, and take one from our Zoom crowd, please, sorry, just.
Hi. Okay. Why do you think the southern resident whales are so skinny?
Yeah. Well, I think it's a combination. We call it death by 1,000 cuts. Are you able to hear it? Yeah, okay. So part of it is that the salmon, specifically the stocks they're interested in, have declined considerably. Not all Chinook stocks are declining, but the ones they're interested in more. This microbiome issue, it could be related to contaminants and inflammatory responses, which is changing the microbiome, which is changing their ability to absorb nutrients. So I think it's multifactorial. And what's even worse in all of that, the synergy between those, is that because you store your contaminants in your blubber layer, what do when you get skinny? You mobilize it. And then when you mobilize it, all those toxins are now circulating through your body. So it's a real, it's a real circular mess of impact. And then, of course, the other thing is the most contaminated, the closer the salmon are to the mouth of the Fraser, the more contaminated they are. Because what happens is, just like the whales, when the salmon are heading to their migratory, to the reds or their breeding, so the area where they're going to lay the eggs, they actually stop feeding and they start to get skinny. So they're losing fat, which means every gram of those fish is higher in contaminants than a big fat healthy one. So it's all of these factors all rolled in together. So the southern residents are more contaminated than the northerns because they eat the fish closer to the mouth of the migratory stream. The killer whales are having to mobilize their stores more, so there's more movement. And the southern resident killer whales have a very low fecundity or calf basically reproductive success. They have a larger intercalf interval. So let's say you would have a calf every 2 or 3 years. Southern residents would be every 4 or 5 years. They have higher neonatal loss. So there's just there's a myriad of factors. So we just call it death by 1,000 cuts. And so it's really hard to say, oh, just if you slow down the ships and make them quieter, everything will be fine. It's very difficult to figure out what the smoking gun is for the southern residents. It's the same question we have. Thanks for asking.
Great, okay, so we'll go back to the audience here for another question.
Thank
Yeah.
you. Thank you. Your talk was wonderful.
Thank you.
Really got
Yes.
some inspiration.
Thank you.
I have a background in fluid mechanics, so I was
Oh,
quite
good.
interested in the number of, inputs you use.
Mm-hmm.
And one thing that interested me was the drag, the energetics of the whale moving through the water.
Yeah.
And I was curious how that gets kind of calibrated, or do you know anything about what's behind that? How it was originally studied and
Yeah, what's really, well, actually, I think this slide actually is one that this is a physicist, Alan Shorter, and so what he did was he used tags with basically measured the velocity moving through water, and then those pucks, those yellow bars are, I don't know if you've ever seen these videos where they either a green screen or kinematics for a tennis player, and they're able to precisely measure movement. And so he was able to look at all the kinematics of these individuals and have them basically run laps, and then look at how that related to the DTAG fluctuations of fin movement And then he could measure the metabolic output of the individual and correlate it back. So he's developing these algorithms that allow you to say, if the dolphin, killer whales are dolphins, if the dolphin does this and moves in this manner, the energetic cost would be this. What's really interesting in what you mentioned is, I did some work with elephant seals, and it's the same thing with a killer whale. Fat killer whale is buoyant. So you don't need to expend a lot of energy to be at the surface to breathe. But if you're a skinny whale, and we've seen this in the whales who are basically, very close to dying, the other whales have to come and lift it up to the surface because they're negatively buoyant. And so buoyancy does play a major factor in how efficient you are, and you kind of want to be neutrally buoyant at the point where you're existing, which is breathing close to the surface. So yeah, it's a great question. Don't have an answer.
Nice. One more there. Just curious, with the kinematic tags, if you noticed any change in behavior or that like locomotion cost of the whales that is associated or possibly correlated with that vessel fact?
Yeah, and that's a really good question. So my colleagues at NOAA, we collaborate a lot. So they're basically the TFO of the States. So NOAA takes the southern residents and I take the northern residents, and then we bring our data together. And because they have acoustic recorders or hydrophones, you can not only measure the vocalization and echolocation of the whales, you can also measure the ambient environment. And what they did was developed a really interesting correlation between foraging success. So you look at things like there's certain kinematic factors that tell you it's a foraging dive. Something called peak jerk. So where there's a rapid change in acceleration. There's a tortuosity or there's roll. These are all kinematic signatures that are associated with foraging dives. And then of course the echolocation clicks, and then they get so close together that they make like a creaking or buzz sound. So rather than going click, 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 you and the crunch. So all of those tell us, oh, that's a foraging dive. And what they did was they looked at the foraging dives and they looked at the ambient noise. And they found, it's a new paper that we just put out called Males Miss and Females Forego. So males will continue to try and forage even if it's a cacophony of sound around them, but they will fail. So they'll do all these foraging dives, but they won't be successful. Females say, I'm not even going to bother. So when the decibels get up to around, I think it was 110 dB in the ambient noise environment, they stop foraging. So it's a really interesting, the first empirical evidence. I mean, from a physics point of view, it's, noise masking is a very real numbers game that you can calculate. But having those D-tags and actually showing that decrease in foraging with the ambient noise environment was so valuable. It's the first empirical evidence that we really have to put on the table and say, look, at noise is really detrimental to these labs.
When you say they ended up doing, they like head back to the search.
For the females just don't even bother foraging.
Oh,
They don't even undertake a foraging dive. The males will go down and forage, but they'll no prey handling sounds, and there's no successful endpoint to that dive.
And in a fairly confined area, that noise level would probably increase as well.
It's so interesting, the differences. We have hydrophones all up and down the killer whale critical habitat, and places like Boundary Pass are actually the quietest when there's no vessel there, because they're almost in a sound shadow. But when a vessel goes in, it's obviously noisy. But places like Swiftsure, which is open to Japan, you get noise coming from, way offshore. It's constantly noisy there. So it doesn't get as quiet as Boundary Pass. So each of those locations has, some have more wind, and it's shocking how noisy rain and snow is. It's shocking how much environmental noise there is. So, they've always had to deal with noise and noisy environments, currents, etc. They all interfere with the echolocation. But, the ships are just, an added insult to, to their existence.
This picture is getting very complicated.
Oh, it is, it's very complex.
Do we want to take a question from our audience then, on Zoom?
Sure. Just wait.
Unless you had an example, were you gonna show something?
No, I just want to put the map up there.
Okay.
So when I'm talking about those different areas, there's La Perouse. And so the recorder at Swiftsure Bank is noisy. The one at Souk is actually quite noisy, just from a anthropogenic, from a natural noise point of view.
Rocks?
Yeah, exactly. So the currents and wind, and it's quite noisy. But then when you go into Boundary Pass, because it's kind of sheltered from outside noises by the islands, it's actually quite quiet when there's no vessels in it. But when the vessels go through it, it's noisier because the noise is contained. So yeah, very complex. I don't do the physical acoustics. We have some great colleagues over on the island that work in that, and I'm just an infant when it comes to my understanding of that. It's highly complex.
Simries?
Simres, yeah, the
Yeah.
Simres is the Turner Island Marine
Research and education.
thank you, Research and Education Society. Yeah, so they have hydrophones, and they also have theodolites, which is a device that allows you to measure distance. So you can look at the distance of a whale and the distance of a vessel. And yeah, they do a lot of good work.
Nice collaboration there.
Yeah, it's great.
Let's go to the online,
Okay.
please.
This is from Joan. Do you do any research on Big's killer whales? They appear to be doing very well despite eating much higher on the food chain
Mhm.
and with a diet that should have a higher level of bioaccumulation.
Absolutely true. They're very high in contaminants, but again, they're not mobilizing. So it's like asbestos, right? If you don't touch it, you're okay. And so they have they've got a good fat layer. They're much more robust animal. If you look at the skeletal structure, they're masseter muscles that, just their jaw is more robust because they're eating marine mammals. And so, yes, they are higher contaminant. Their biopsies and their lipid samples are higher in contaminants than the southern residents. But again, unless you mobilize it, it's not going to affect you. And so that it stays in the blubber layer, and they're doing incredibly well. You're right. We see them much more frequently in the inner waters. And this is one of our questions, okay, the southern residents aren't coming in and the bigs are. It's so interesting. I've been on the water when northern and southern residents are going by in Johnstone Strait, which is not very wide. And we're listening on the hydrophone and you can hear them both and suddenly it's dead silence. They sort of pass each other. And then they start calling again. And there's almost an avoidance between these different groups. So that was one of the factors that we looked at. We're just working up were there factors that drove the southern residents out? I.e., is it noisier? Is there more vessel traffic? Is it the big whales coming in? Or are the factors, it was the push me, pull me. Are they being pushed out or are they being pulled out? And it turns out it looks like they're being pulled out by better foraging conditions on the outer waters rather than being driven out by, let's say, noise or maybe the presence of the bigs. So, we looked at a lot of those factors, but the one that really seems to be driving things is the salmon. So yeah, I do a little bit of work on the bigs, but because they're doing well, I always get the problem children. So I focused on the whales that are doing as well. And so I'm working on the southern. Dr. Thomas Donnell Velcroze over at Pacific Biological Station does a lot of work on the bigs. Yeah, but they're certainly fat and healthy. The one thing I will mention is that even if a whale is fat and has a good lipid layer, when it has a calf, a lot of those lipids are mobilized for lactation. And so that first calf, unfortunately, gets the benefit of its entire contaminants load from its first, 15 years of life. And then that mother will have less contaminants and start building them up again. So the next calf will get it. So that's one of the detriments for that intercalf interval as well. It means that when you do have a calf and it's been 5 or 6 years since you lactated, that calf is getting a higher load, like almost like the first calf. So there's it's very complicated, but that's a really interesting aspect to the populations as well.
Yes.
Oh, yeah, a question about, you were talking about how the prey share.
Mhm.
But, the question I have is about the juvenile whales, at what age do they start kind of actually participating in, foraging as opposed to just kind of taking, and is it different between the different, populations, like
Yes.
easier to learn to hunt seals and salmon?
Yeah, I mean, they start this is more into behavioral ecology, which is, I depend on my colleagues for this kind of information, so I'm not speaking from a place of firsthand knowledge here. But certainly the bigs, you see them playing, like they'll be playing with they'll even play with birds and things, just like a cat, and they'll learn a lot of those techniques. Sometimes they might provide a dead seal for them to throw, because I don't know if you've seen, it's very percussive. They'll come down and percuss to stun a faucid or an odoride, and then they'll pick it up and they'll throw it in the air. And so you see the young ones, just like any other species, playing around with that. With respect to the southern residents, because of that longer intercalf interval, sometimes those calves will stay and continue to nurse on the mom maybe longer than what would have happened if another calf were born. But I actually don't know the age at which they would start being 100% capable of foraging on their own. It's a really good question. I'll have to look that one up. Nope, we're all good. Oh, one more here.
You mentioned the flexibility in diet, because from read, I don't read scientific articles, but from reading news, they seem to say that southern resident killer whales eat exclusively Chinook salmon and will turn out their nose at other species. But you mentioned sablefish.
Yeah, so they do, it really is, it's 70 to 95% of their diet in the summer does appear to be Chinook, and it makes sense. Those are the fattiest, richest fish. The sablefish might be more costly to forage on because they're very deep. They might be more diffuse, whereas when you're dealing with a funnel where all the fish are coming in, it's a great place to sit and forage. So it makes sense why they would focus on Chinook in those summer months. And as those Chinook populations, which to me is as a metabolic biochemist, I'm blown away by salmon, that you can have a fish who will leave a stream, go out to sea for 4 years, know exactly how to come back, and come back within around 4 days of the peak of that particular stock. How did they do that? I mean, your whales are interesting, but the fish are amazing, that they're able to do this. So the migratory timing windows are pretty tight. So they can sort of sit and as those migratory timing windows start to maybe spread out a bit, they might start supplementing with Chinook and coho, who would then those runs start in the fall. So that's when we see them move from Swisher. So they're coming in from Swisher and going down into Puget Sound, where they start to feed a bit more on coho and Chinook, because those runs are available. So they do really, they do time their dietary focus on what's available and what is really the most energetically valuable fish. But when you look at the energetic content of a Chinook compared to a coho, it's like a quarter. So you'd have a lot more foraging effort to get the same caloric value. So it makes sense what they're doing. It just and they don't eat, but what doesn't make sense, they don't eat sockeye.
They're quite fat.
Yeah, not as high lipid content. Chinook are the highest. And so they don't eat sockeye. You know, maybe you might get the odd genetic blip in the fecal saying, yeah, they ate a sockeye or yeah, they ate a pink, but you know, that's not really, it's coho, Chinook and chum.
And are the sablefish only sort of from kind of that white line this side of Swisher Bay out? Or are there sablefish in the,
Just the young ones from what I understand. So a lot of this, I mean, we've really been digging to try and find more information on the sablefish. But if you look out of Port Renfrew, there's a First Nations fish plant there. And so in talking with Apache Dat, there's been a few years where the halibut fishers are coming in and just complaining bitterly because they can't even get their gear to the bottom before the sablefish hit it. And so, you know, there's been sort of, I guess, some waves that have been coming through. From what I've been speaking with the fish folks over at PBS, there's more understanding coming through on the sablefish. But because the fishery is where we get most of our information
and the fishery is out.
Is out there, yeah. So we actually don't really have a lot of knowledge on you know, how those young move. You know, and you know, when I first started and asked about salmon, because to me it's the Department of Salmon and Oceans, because there's so much research on salmon going on. And they say, yeah, we know everything about salmon. I said, well, where do they go? Where are they? Or should they go out there? And no, it's not very helpful. So it's really been Dr. Cam Freshwater has been, it's 5 years. He's been with DFO and I've been working with him and he's phenomenal. And he's been doing things like putting tags in the fish. So he'll go up at Bamfield, Port Renfrew, and you tag the fish. And then you basically lay down a net of recorders. And so when the fish goes by, it picks up the signal from the tag and it pings. And so he's able to track these fish movements. It's phenomenal stuff. So we want to try and overlay those fish movements with the killer whale foraging movements and get a better understanding of all of that. But somebody needs to do that with sablefish because we don't know much at all. They're fascinating, but I don't really have much knowledge.
So, and when you said that the southern residents are moving kind of more offshore, not offshore like this.
Yeah, a little less inland
So
waters, yeah.
It's not that there is more of them that live out there, it's that they're spending more of the same population is spending
Yeah.
more time out there.
Yeah.
It's not
So we only have
that there's
74
more
whales.
of them.
And so we see them on Swiftsure, and when we look at the acoustic, we have an acoustic recorder that was put in 2009. And then we put ours in 2018. So we had a decadal difference between our recorders, and it really didn't change much. We saw the same, you know, frequency of occurrence. A little bit later in our research in the later decade, because the fish are migrating a little bit later, so everything made sense. But what they weren't doing is they weren't going into San Juan and San Juan Island down in Friday Harbor around that area. Fantastic. And people, you know, like Orca Island, right? Like they live and breathe orcas there. They all have homes along. They've got everything is orca, and they're just not coming in anymore. They're coming in really late. And they're only coming in, you know, there was, I think the worst year was only 17 days over the entire season, like 4.5 months or something. It was really low. It used to be like every day, every second day. There was guaranteed, you know, that somebody would see one in that area. And so it's been a real shift there's only 74 whales, right? So it's the same whales. And when we see them out at Swiftsure, and now all the researchers who did all this phenomenal work there are coming out to Swiftsure with us, and we're seeing more researchers on Swiftsure because we get all the whales.
But the population it's not like having moved out there, they're obviously doing a lot better. It's just that they're surviving, where maybe if they had just stayed on the inside, they would have been even more precipitous.
Yeah, it's really hard to say because you know, if the Columbia fish, so if you go down the coast, so in the winter, as we turn to spring, those Columbia runs are sort of their first, you know, big meal after a pretty lean winter, and so they stay around the Columbia. River for quite a bit, and they'll pop up to Swiftsure and see how the fishing is and go back to the Columbia. And then gradually they'll start coming in and increasing in what we call intensity of occurrence. Starts sort of in May and in June, and then peaks in July and August and starts to drop off in September. So they really stick right around Swiftsure, and then used to really go in. So they'd come down through Juan de Fuca and really not do too much foraging in Juan de Fuca. They would forage on Swiftsure, and then they'd move down Juan de Fuca until they got to, I don't know if you can see, there's Harrow Strait, and then you can see by the yellow dot there right here. This is San Juan and Harrow Strait, and this is quite a sheer wall. And it's just a fantastic foraging environment. And it's so funny because it's the sort of American side of the strait is so much foraging that goes on, but the Canadian side of the strait is a very different bathymetry, and they don't really forage on our side. So the boundary goes right down the middle of Harrow. And so that's really the American side is a key foraging area. And the fish are still there, but the whales are seeming to choose outside waters a little more.
So I'm going to, I hate to slow this down a bit because I think we have more questions. I certainly have a few myself, but I think you've got your work cut out for you, have had it cut out for you for quite some time. So I really appreciate, I hope everyone else has appreciated this very interesting and just full on, so much information, so many interconnecting things that are happening that can be affecting the whales that are more than what we probably are thinking when we listen to the news, that there is just much more complicated than,
Absolutely.
and not so easy to draw these conclusions and regulations, that sort of thing. So I'd like to thank you very much for that. If everybody wants to give a. And before we close off on our Zoom, I wanted to thank the marine biology section. Jen is here, a new Jen maybe, and Deb here as well. And thank you very much, Frederick, for helping out on the IT. That's been much appreciated. And this is our last marine biology section for the spring seasons, or for the summer, I guess I would say. But do please return for the AGM coming up soon, and all those other events that you can access at naturevancouver.ca. Thank you very much, everyone, for coming.
Thank you very much. Yeah. There you go.